Hi, everyone, and thanks for joining us today. I'm Dr. Kelly Deal, the Senior Director of Science Communication here at Morris Animal Foundation, and I think we'll get things kind of moving along. We've got a really full agenda. Again, thanks for everyone who is joining us today for kind of part 2 of our canine cancer series. These folks will be introducing themselves, but we'll be hearing from Dr. Dave Bruyette, CMO of Anivive Lifesciences, and Dr. Heather Wilson-Robles of Ethos Discovery and the Oncology Service, and both are Morris Animal Foundation researchers. We'll start with Dr. Bruyette's video, followed by Dr. Wilson-Robles. You guys should be seeing right now the welcome slide with the housekeeping rules. Just to go over these again, we'll have a live Q&A session at the end of the webinar, but if you have questions that crop up as the videos are playing, please put them in the Q&A function. Obviously, you can reach out to us for tech issues, comments via the chat, and closed captioning again is enabled for this session if you need it. So why don't we go ahead and start the first video?
Hi, everybody. It's great to see you today. My name is Dave Bruyette. I'm the Chief Medical Officer at Anivive Lifesciences, and I'm here today to talk to you a little bit about our company, Anivive Lifesciences, and how and why we got involved in veterinary oncology drug development. So I'm going to go ahead and share my screen, and we will go from there. So from the very, highest level, why do we care about veterinary oncology? And I think we care because all of us are at risk, pet owners as well as our pets. So we know that there's over 1.6 million people diagnosed with cancer every year in the United States. But you may be surprised to know that more than 4.2 million dogs are also being diagnosed with cancer. And the cancers that are occurring in dogs are very similar to the cancers that are occurring in people. These are naturally occurring tumors. And their presentation in animals, the pathophysiology behind how they get those cancers, response to treatment, are very similar to what is seen in humans. So as a result of that, humans are a good animal model for dogs, and dogs and cats are good animal models for humans. And especially when we look at some of the more sophisticated analyses of these tumors, like the genetic makeup of these tumors, we see that there's quite a lot of similarities. So when we look at man, we see about 28% of the cancers occurring in men are prostate, and then lung and colon and rectum make up the other two common tumors. In women, not surprisingly, breast cancer is very high in incidence, again, followed behind lung and colon and rectal cancers. When we look at dogs, though, obviously the most common cancer that we're concerned about is lymphoma, and then adenocarcinomas in various tissues. Osteosarcoma is obviously an interest to the Morris Animal Foundation and others. And then uroepithelial tumors and mast cell tumors make up probably the most common tumor seen in veterinary practice. Now, for the purposes of this talk, one of the things we want to discuss is, well, what's the path to get a drug approved, either for use in humans or for use in veterinary medicine? So at the top, this is sort of the traditional pathway for human drugs going through the FDA. All therapeutics for humans, whether they're small molecules or biologic like monoclonal antibody, all go through the FDA for approval. And the time frame from when a basic researcher discovers one of these molecules till you get an approval for that molecule is a little over 14 years. And on some of the most recent estimates, that can cost a pharmaceutical company upwards of $2.8 billion to get the drug approved. One of the staggering things about this, in addition to the timeline and the amount of money involved, is that in the oncology space, 90% of the drugs that start down this pathway end up in failure. We'll talk about why that happens and ways that we at Anivive are trying to reduce the risk of failure as we go down the path of oncology drug development. On the veterinary side, it's a little bit different. All small molecule treatments all go through FDA, similar to human, whereas biologics like monoclonal antibodies are going to go through the USDA. But to compare apples to apples, if we look at the timeline and the amount of money spent to get a small molecule through the FDA, again, from start of discovery all the way through to approval, is going to be somewhere around 12 years, and roughly $100 million will be spent on getting that product all the way through. Now, when we first started the company and we were looking at wanting to get into the oncology space, we wanted to make sure that we were getting into the oncology space in a very intelligent and thoughtful manner. And we wanted to build upon the successes as well as the failures of those who preceded us. So we adopted fairly early on a couple of strategies that we think have gone a long way towards helping us de-risk drug development. One of these was called the 5R framework. And this is looking at a therapeutic in an agnostic way and answering some basic questions before you start down the development path. The first being, do you have the right target? So does your drug interact with the protein or the gene that you're trying to impact that you think is driving that particular cancer? Does your drug actually get to where it's going? So this is where pharmacokinetics and pharmacodynamics and things like tissue exposure come into play. Because if your drug doesn't get to the target, it's not going to be very effective. Is your drug safe? And we'll talk about this when we talk about how we choose drugs on the veterinary side. But these drugs need to have a very high safety margin. We need to understand the interaction between this drug and other drugs that the patient may be taking. We want to recognize, well, what is the patient population? Who is the right patient for your drug? And this is based on the type of cancer that may be present, the response rate that those dogs and people have historically had to other treatments. And at the end of the day, if you're a drug company developing these compounds, you want to know, well, does this make sense from a commercial perspective? So is the intellectual property or the patents, are these things all protective, and do they have a long time frame so that you can work to recoup the cost of these drug development stages that we discussed earlier? By adopting this 5R framework, one of the companies that has been quite successful on the human side is AstraZeneca. And this is sort of displayed on the right-hand side of the slide. And basically, by adopting this 5R strategy versus what they did previously, they were able to maximally increase the percentage of successful projects, whether they were preclinical, phase 1, phase 2, or phase 3 projects. And more importantly, and again, this is talking specifically about oncology drugs, they increased their success rate from 4% to 19% just by simply adopting this policy before they started their development pathway. And when you look at reasons why these drugs failed going through the prior iteration and why they were successful after adoption of the 5R therapeutic cascade, is that you saw far fewer drugs now failing as a result of problems with strategy or problems with safety or problems with dose distribution. The biggest problem that human pharma companies still have is inefficacy. And that's because they tend to go from in vitro testing to rodent models, and then oftentimes may skip straight to humans, bypassing an important step in the pathway, which we think is evaluating these therapeutics in pet dogs and cats. Looking at project outcomes, again, why are projects successful and why do projects fail in the oncology space? Again, adopting sort of this 5R paradigm. AstraZeneca was able to demonstrate that their success rate went up from 23 to 48%. The cycle times, how long it took to go through the FDA, decreased dramatically. And the reason for project closure started to change, and again, eliminated things like problems with strategy, problems with not being able to identify correct leads, which again helps them de-risk the project. And again, more importantly, when you look at the success rate and you look at the percentage of projects that close versus the percentage of projects that continue to go on, they saw a significantly improved pathway simply by adopting a more rigid criteria from the get-go. Now, this path can also work bidirectionally. So we've seen not only using human drugs as an example for developing veterinary drugs, but veterinary drugs going the other direction back into people. And there's numerous examples with a variety of different mechanisms of actions in terms of the therapeutics, where data generated in pet dogs was then used to further accelerate development of human drugs undergoing development. So when we started and we said, okay, we want to be in the oncology space, the question that came up immediately is, well, we understand that, but why lymphoma? Why are you going after lymphoma as your first drug and your first target? Well, as an internist and as a non-oncologist, I had spent 25 years of my life referring cases of lymphoma over to the oncologist, and really not much had changed in those 25 years. We still had these common issues facing pet owners and veterinarians. With respect to lymphoma, roughly 80% of pet owners were opting out of treatment and electing only palliative care, despite the fact that we knew that lymphoma was the most common hematopoietic tumor in dogs, that more than 2,000 dogs are being diagnosed with lymphoma every day. And despite evidence from insurance databases that lymphoma was amongst the most expensive and most common medical claim that was being placed by pet owners. So what are the reasons why 80% of people opt out? Well, some opt out because they just simply can't do it. They can't make it to an oncology specialty practices, especially for injectable chemotherapies. They're oftentimes very afraid of chemotherapy because they're really worried about side effects. And this is also true of referring veterinarians. They don't want to do anything to decrease the pet's quality of life as they're undergoing treatment. There's also concerns about cost. So the standard of care CHOP-based protocol for lymphoma in dogs, depending on what part of the country you're in, can run anywhere from $6,000 to $10,000, which can be a huge barrier to people adopting treatment. So pretty much early on, we listened to Cheryl London, who said, you know, you really need to do something, whatever your drugs are, whatever drugs you're thinking about, they need to be able to help mitigate these concerns. So if we look at the number of dogs being diagnosed with cancer every year, being diagnosed with lymphoma, of those 700,000 cases, only about 155,000 are being treated with CHOP-based protocol. About 85,000 are getting full-blown CHOP. Another 70,000 or so dogs might receive single-agent chemotherapy like doxorubicin. And all of those dogs would certainly be eligible for additional therapeutics. But the bigger unmet need is these 550,000 dogs who are receiving no therapy at all. So we decided to kind of take a different approach, and that is, rather than trying to repurpose something that was being passed on to us by the founders of the company, what we decided to do was to take the 5 R's into consideration and also use software, because we are a software-driven company. Well before AI was the sexy thing to talk about, half of our company is devised of software engineers. And we sat down and developed what we wanted to do with a given cancer and a given therapeutic. So we developed very specific target product profiles to help tailor the ideal therapy up front, and then go find the therapy that best falls into those categories. So with respect to lymphoma, we wanted something that would be active in both newly diagnosed and relapse cases. We wanted something that had a novel mechanism of action, because if oncologists wanted to use the new drug in conjunction with standard therapy, we wanted to make sure that it made sense mechanistically that those drugs could be used in combination. We also wanted something that was safe, either as a single agent or in combination. We didn't want to see overlapping toxicities, which would preclude its use. Importantly, we wanted to make sure that the drug or drugs would maintain quality of life. And that was one of the things that Cheryl looked at as she was working on the drug that we'll talk about here in a second, which is verdinexor. And we know that multiple drug resistance caused by the multiple drug resistance gene is a common reason for treatment failures in veterinary oncology. So we wanted a therapeutic that did not interact with or serve as a substrate for the MDR gene. And all of that all sounds beautiful, but how do you actually do that? You know, there's more than 8,000 chemicals being synthesized every year to be used in the oncology space. It's not physically possible to vet through all of those. So the software team developed a series of algorithms, which we continue to optimize, which helps us deal with the huge amount of data that comes in on a daily basis in the oncology space, as well as in our other drug development candidates. So we compile all of these different databases. Some of these databases are proprietary. Some of those are databases that are publicly available. And we start to sift through those 8,000 compounds, lining them up with what we thought we're looking for in our target product profile. So you very quickly can narrow down from 8,000 compounds to 20 to 30 compounds. The next thing we want to do, especially in the oncology space, because oncology is essentially a disease of your genes gone bad, is that we wanted to know, well, look, in these certain cancers, and particularly with respect to lymphoma for this conversation, are the genes that are active in canine lymphomas and the genes that are active in human lymphomas, are they the same? And if they are, that serves as a good marker that if our drug interacts with the gene or the product of that gene, that de-risks that product and moves that candidate higher up into the list. We then start to look at other databases like intellectual property databases to look and make sure that the compound that we're looking at does have patent protection. And eventually we're able to bring that down from 8,000 to 30 to 10 to 3 or 4. And then we vet through those, start to reach out to those organizations who are developing those compounds and look to see if there's an opportunity to bring it forward on the veterinary side. So the first drug that we were successful doing this for in the oncology space was Verdinexor, a SINE inhibitor being developed by a human biotech company in Boston known as Karyopharm. They were bringing forward a drug, Selinexor, the sister molecule of Verdinexor, and we were able to leverage a lot of the work that was done with both Selinexor and Verdinexor at Karyopharm to help shorten the time to approval and decrease the expense involved, because when we get the drug to market, we want to be able to bring all of our therapeutics to market at a price point, again, where owners would be able to opt into treatment and not opt out of therapy. So the first SINE inhibitor that was approved was Karyopharm's drug Selinexor, originally approved for multiple myeloma, but subsequently now also has an approval for B-cell lymphomas. So while they received the first approval, the second approval of a SINE inhibitor was Anivive, and that was the approval that we received conditionally for Laverdia and the treatment of lymphoma in dogs. Again, this was specifically, we went after many of the attributes that Laverdia has. It's oral, ease of administration, good safety profile, good quality of life for the dogs who are taking the drug, different mechanism of action, and does not interact with the multiple drug resistance gene. We were also able to bring it forward at a price point to the vet where the veterinarians would be paying about $100 a month for every patient, and then they would pass that on for an additional markup to the pet owner. But again, at a price that's significantly less than what owners would normally be paying for traditional cytotoxic chemotherapies. So shortly after licensing or getting the approval from the FDA, we made an arrangement with Dechra Pharmaceuticals, where we out-licensed the commercialization and marketing rights to Laverdia. This allowed Dechra to go ahead and utilize their extensive sales force, as well as their capabilities in other countries, to help bring Laverdia forward, and again, with the goal of treating as many dogs as possible with lymphoma. Anivive is still involved with research activities related to Laverdia, looking at other novel indications for SINE inhibitors. For instance, we're looking at hemangiosarcoma and osteosarcoma. We're looking at doing some PK studies in cats to see if the drug will have a good safety profile in cats. But we're going to continue to look at other therapeutic indications for the drug to help increase the usage again in canine cancer. The efficacy that we submitted to the FDA resulted in a granting of a reasonable expectation of efficacy. We're currently in the end stages of our pivotal field study for full approval, looking to target that study by the end of the year. The only real difference between a conditionally approved and a fully approved drug is that for a conditionally approved drug, you demonstrate reasonable expectation of efficacy, but the safety and the manufacturing criteria are identical to those of a fully approved drug. So we're not looking, and we were never looking to have Laverdia viewed as a replacement for CHOP. That's not how the drug should be used. It's really just a way to optimize access to care for veterinary patients who have lymphoma. And now we're building a sort of a collaborative consultation network here, where you have the patient, the canine in this case, being diagnosed with lymphoma. working very closely with their primary care vet and the pet owner, and then seeking input from a specialist to make sure that every single patient is given the best possible care tailored for that patient and for that client. Now, using that same platform, we're also looking at other cancers, osteosarcoma being one, and then the therapeutic that will probably be the second oncology asset that we bring to market will be a bispecific angiotoxin for hemangiosarcoma in dogs. Obviously, this is a very common cancer affecting dogs. Standard of care historically has been splenectomy plus traditional chemo. Again, very similar to lymphoma, not much had changed over the last 40 years. And unfortunately, the survival times have also not changed with that disease. We were able to, again, go through the databases and through the Anivive Select process and ended up licensing EBAT, the specific angiotoxin from the University of Minnesota. This compound not only targets abnormal cancer tissue, but spares normal tissue because in order to release the toxin, 2 different proteins have to be present. And when you look at the data that they published in peer-reviewed journals, looking at dosing EBAT in conjunction with surgery and standard of care chemotherapy, we saw marked increases in 6-month survival and 12-month survival in dogs. And many of the dogs, 25% or so of the dogs, were still alive at more than 450 days after treatment. So we think that this therapeutic, which right now is pending, the efficacy technical section right now is on file and being reviewed by the FDA. We hope to have that by the end of the year. And that will allow us to accelerate our development program on the manufacturing side so that we can then bring EBAT forward into the veterinary space. Again, the data was quite compelling in the studies done by Dr. Modiano and Dr. Borgatti at the University of Minnesota, showing in 2 different studies the efficacy and safety profile of the drug, and helped us determine the starting dose, the effective dose, and the efficacy and safety signal that we see in pet dogs with spontaneous hemangiosarcoma. Other things in our pipeline, obviously, we're going to be looking at other therapeutics for oncology, osteosarcoma being the next one on the list, and we've got some exciting leads here, both on the therapeutic side as well as the therapeutic vaccine side. We're also using these same strategies for our antifungal platform, for valley fever, for histo and blasto, for gingivitis. And then we have some novel compounds that we'll be looking at for longevity and life extension in pet dogs. So I'm excited to have been here today. Be happy to answer any questions that you guys might have during the open Q&A that we'll be having here shortly. You can always reach me at Anivive. My email is david@anivive.com, and I'd be happy to talk. So thanks again for your time and look forward to the rest of the session.
Thanks so much, Dave. I don't know for everyone listening, but I was especially interested in the lymphoma. I don't think you get gray hair like me and have had pets for 50 years without encountering it, not only in my patients, but I had a dog with GI lymphoma and even worse, a cat with cutaneous lymphoma, which happened to be my first cat that I actually owned. And it was really heartbreaking to lose her. So I'm really, really excited about these developments. Now we're going to hear, as I mentioned before, from Dr. Heather Wilson-Robles, and I'm excited to hear what Heather has to say too.
Good morning. My name is Heather Wilson-Robles. I'm a medical oncologist with Ethos Discovery, and I'm excited to talk to you this morning about the research that we're doing and how it may help golden retrievers. So you might ask, who is Ethos Discovery? I've never heard of this before. And we're a pretty small organization, so it wouldn't surprise me if you hadn't heard of us. But we're a nonprofit incubator of scientific innovation. And what does that mean? That means basically we try team science. We take some really smart people from all different areas of expertise. We put them together and we really troubleshoot and try to figure out what are the biggest problems and how can we take them on and try to solve them. We have 7 or 6 main areas of interest and focus that we work in. Oncology is probably our biggest area, and the main reason for that is we are a little oncologist heavy, so we have more oncologists than other areas, but we also have internal medicine specialists with interest in gastroenterology and the microbiome, as well as interest in the kidneys. We have criticalists with interest in emergency critical care, mostly sepsis, so those systemic infections that make people and animals very, very sick. We have boarded surgeons and surgical oncologists who are interested in innovations in surgery, and we'll talk a little more about that coming up, as well as neurologists interested in neuroinflammatory diseases, things like Pug's dog encephalitis, and then new therapeutics and diagnostics, and so making new drugs or reutilizing old drugs in new ways. So this is a slide that just sort of shows some of our clinical trials in our different areas. And so in oncology, we've got 3 current trials that are enrolling. I'm going to talk mostly about Ethos Push today, but we also have drug trials with Verdinexor, incorporating it into a CHOP-based chemotherapy protocol for dogs with lymphoma, and a new investigational drug, AOH 1996. In surgery, we have Mimic, which again, I'm going to talk about a bit today, as well as Capcom, which is a drug or a study to look at removing or treating bone tumors without having to amputate that leg. In our gastroenterology section, we have studies that are really focused on the microbiome. So Del Taco, Del hemangiosarcoma, and metabolomic studies, really looking at how the microbiome affects disease, affects metastasis, affects disease response, drug resistance, those types of things. In critical care, most of our work is focused on sepsis or these sort of systemic infections that make animals and humans very, very sick. In neurology, we're looking at those inflammatory brain diseases, things like pug dog encephalitis, but also things like intervertebral disc disease in French bulldogs and pit bulls, certain dogs that tend to have extra bleeding associated with their disc disease that gives them a worse prognosis and outcome. And then in our therapeutics areas, we're looking at new versions of rapamycin, so Rapaboil or Rapanano. These are new formulations that may allow us to use these drugs more successfully, allow them to get into the bloodstream and be more active than their current formulations. So I'm going to start and talk to you guys about Mimic. Now, Mimic is our new study that is looking at minimally invasive metastatectomies in dogs with osteosarcoma and other pulmonary tumors. This is being headed by Chris Thompson. So he is one of our board certified surgeons. He's also a surgical oncologist and has a practice in Southern California. The Mimic trial really is looking at removing the spread of bone tumors that go to the lungs. So primary bone tumors, these happen in big dogs, dogs like golden retrievers. They often happen in their legs, in their limbs, front legs or back legs. And we usually treat that with amputation, and then we follow with chemotherapy. The same as in humans. They'll often treat those with amputation of the limb or what we call limb sparing, removing part of the bone and putting in a prosthesis, and then following with chemotherapy as well. In humans, this is a disease that's common in children and then people over 60. And in dogs, we see this in large and giant breed dogs. In humans, often when they develop spread of this tumor to the lungs, which happens 90% of the time in dogs and maybe a little bit less in people because they're more intense with their chemo, but the most common site for it to go is the lungs. So in dogs, most of the time we say that's the end of their journey. We may try some chemo or try a few other things, but most of it's palliative care and hospice care once we find the tumor in their lungs. But for humans, their journey goes on a little further. And in many cases, they will have those lung tumors removed. And we have seen that there's a substantial benefit to those patients who do undergo surgery for that spread. And we don't do this often in dogs, and we wonder why. And I think one of the main reasons is this idea that you don't want to put this older large breed dog who's probably at the end of their disease state through a big surgery that's going to take them a long time to recover from. And so we thought, well, if we can remove those barriers and make this a quick surgery that's easy for them to recover from, that they can bounce back in 1 to 2 days and be home, could it be worth it to then try to incorporate this type of therapy into our standard osteosarcoma care for dogs. So the benefit that people derive from this tumor or from this type of procedure is quite stark. So a study in 2005 looked at over 1,700 patients with osteosarcoma that had spread to their lungs. And what they found was that those people who had their tumors removed, 40% of them were still alive at 5 years, whereas none of the people who didn't go to surgery were still alive at 5 years. And what they found was that when this was your very first recurrence, so the primary bone tumor has been removed, you've been fine, and then all of a sudden we find stuff in the lungs, the very first time, still 2/3 of those people went into a complete remission after their surgery. And so those were the guys who were most likely to then go on and be cured or have that long-term survival. Now, in dogs, there is a paucity of literature out there. Probably the paper that we all look to the most is one by Turner that was published in 2017. They looked at 194 dogs, so nearly 200 dogs with osteosarcoma that had spread to the lungs. Now, not all of those dogs went for surgery, but some of them did. And what they found was that the dogs that went to surgery had an average survival time of about 230 days, compared to those dogs that didn't go to surgery, where their survival time was just under 50 days. So pretty big difference, 4 or 5 times as long if you went to surgery. Now, what they also found is those dogs who were the best candidates for surgery, meaning they had few nodules, so 3 or fewer pulmonary nodules or lung nodules, and they had gone the longest amount of time between their original surgery to remove their bone tumor and this showing up of and the discovery of spread to their lungs. That was over 275 days. Those guys could live almost another year without recurrence versus dogs who didn't have surgery, which only lived about 100 days. So again, we do see this inkling of benefit in dogs, but we really haven't applied this into our everyday practice to really know how this works. So our main goal was to remove some of the barriers for getting animals to surgery. And we decided to use video-assisted thoracoscopic surgery. And so this is a minimally invasive technique, very similar to some of the things we see in humans, where they're not cracking the chest, they're making small little ports and they're going in with cameras to remove those masses and pull them out. Well, our goal was to have these animals discharged from the hospital and home in less than 40 hours. So right now we have a prospective trial. We're recruiting 36 dogs to Chris Thompson's practice in Southern California. And our goal is to remove those masses and follow them out. Right now, we're trying to recruit mostly dogs with osteosarcoma, although we will take dogs with other lung tumors. So we start with a CT scan that will show us where the pulmonary nodules are. So a CAT scan that basically gives us a full picture of the lungs to know where the nodules are. And then we inject the dogs with a green dye that will allow the surgeon, when they're in there with their cameras, to see those using infrared mapping. So near infrared lighting, those tumors will light up and glow so that it's easier for the surgeon to find when they're in there with those tiny cameras. And then we'll remove those masses, either by removing the lung entirely if need be, or by taking just a piece of that lung, depending on how big the nodule is. So here is a picture of the instrument inside removing a nodule from the lung. Once we've gotten them through surgery, we get them back for regular rechecks and x-rays. We do CT scans at 3 months, 6 months, and then a year. And then we look at the tumors. We get biopsies on every tumor we take out, and then we share tissue and blood with our collaborators. So the collaborators we're working with on this particular study include, of course, our veterinary specialty hospitals with Ethos, but also the Dana-Farber Cancer Institute, the Harvard Medical School, James Cancer Network, Ohio State, Nationwide Children's, and Case Western. And these guys are all receiving blood or tissue or both from these cases. And we're looking at a variety of things. We want none of this to go to waste. We want to gain as much information as possible by including as many collaborators and sharing as much data as we can. And so we're looking at genomic factors, what makes the metastatic lesion different, drug resistance, what is special about that environment that allows those tumors to grow there, markers of inflammation, are there ways to limit the inflammatory response and stop those tumors from growing? We're looking at the blood for markers of disease that can help us track and identify when animals and people are going to have metastasis before we can even see it, and looking for new targets to develop new drugs to better treat this disease. So far, we've enrolled 10 patients. 4 of those have osteosarcoma, and we've removed 33 nodules from those 10 patients. 8 of those 10 patients have been discharged within 24 hours of surgery. So the next day, they're going home. 2 dogs, one ended up being in the hospital for 48 hours. The other one, unfortunately, developed a surgical complication and was in the hospital for 5 days. Currently, we have 8 of 10 dogs who are alive after going to surgery. Their range is 1 to 19 months. The 2 dogs that have had progressive disease had progressive disease at their 6th-month CT scans. One of those dogs had osteosarcoma and the other had malignant melanoma. And so we're continuing to follow these dogs out, and we're continuing to enroll. Now, hemangiosarcoma, of course, you guys are probably also familiar with this disease. This is a tumor that is a tumor of blood vessels. And unlike people or cats, this tumor in dogs tends to occur in the visceral organs, meaning those inside your body. So the spleen, the liver, the heart. We do sometimes see it in the skin or the subcutis or even other places like bone. But the vast majority of these start in the spleen, the liver, and the heart. Unlike people, again, the visceral organs or internal organs are the predominant site, and we usually see about 50,000 cases per year in the US. Typically, when we treat these, we start with a splenectomy. So that means removing the spleen, which is by far the most common primary site or starting site of this tumor. A lot of those dogs will present to the emergency room. They're collapsed, they've got a belly full of fluid, which is blood because the tumor ruptured and bled, and it's an emergent situation. So we usually get them to the hospital while it's an emergency, and then we take them to emergency surgery, not knowing if this is a benign or a malignant tumor. And then once we get that diagnosis, we follow up with chemotherapy. So the poor people who own these stocks are really stuck in this sort of non-expected last-minute emergent situation, trying to decide to take their animal to surgery, not knowing what their prognosis is. This disease is a very aggressive disease, and the 1-year survival rate is really around 12%, which is really pretty terrible. And so our goal is to improve upon that and get that to be much better than it currently is. So how are we doing that? We're doing that with our PUSH study. And I'll tell you what that acronym means in just a minute, but we've basically launched a 400-dog randomized prospective trial. So what that means is we're not looking at the medical records of dogs after they've come in. We are actively enrolling these dogs. We're following them from the beginning, and that's really important. These dogs, once they're found to have a splenic tumor that has bled, that hasn't spread anywhere else in their body, those guys get randomized into one of 4 treatment groups. The first is doxorubicin alone. Now, doxorubicin is our standard of care for this particular disease. And so that's our #1 control group. Number 2 is standard of care with rapamycin, which is that new drug I was telling you that we're working on with Rapaboil or Rapanano. Group 3 is doxorubicin with sorafenib, which is a small molecule inhibitor. It targets specific growth factor receptors, so things that make those cells grow uncontrollably. And then a 4th group for Araxol, which is a novel drug aimed at these specific tumor cells. So because this is a randomized prospective study, you'd be surprised to know, but in veterinary medicine, we don't do a lot of prospective studies. In most cases, the data is all gathered from looking back at medical records kind of in hindsight. And that's really gives us really limited data. Not all the dogs are treated the same. They're not all followed the same. They're not all staged the same. They're not all given the same dose. And so because so many variables are not controlled in those studies where we just go back and look at records after the animals have been treated, we miss things that we can't find because there's too many variables. When you do a prospective trial, you control for a lot of that noise, you control for a lot of those variables, and it gives us a way to understand the disease better and maybe be able to figure out what is it that allows those guys, those 12% of dogs who do become long-term survivors, what is it that's different about them, and can we capitalize on that? we know that dogs who have hemangiosarcoma, even though we say that the median survival time or the average survival time is around 6 months, we know that the vast majority of them will have evidence of spread. So that tumor will spread to the lungs or a lymph node or the liver, some other spot within about 4 to 5 months. 80% of them will usually have evidence of spread by that time. So we know that making it to 6 months is a big milestone. And if you can make it to 6 months, then chances are you're going to make it longer. If you make it to 6 months without evidence of spread, you're probably going to make it a lot longer. So the goal of this study, this is the Ethos Precision Medicine Umbrella Study for hemangiosarcoma. I know that's a mouthful, but this particular study, the goal is to take those dogs who present to the emergency room, okay, and they've got blood in their belly and they've got a splenic mass. We know that they don't have spread of that tumor to anywhere else in their body. We're going to remove that spleen. We're going to take blood samples, and we're going to give those samples and blood to all of our collaborators to just learn as much about this disease as we possibly can. And then those dogs who don't have evidence of spread, who do have hemangiosarcoma, are going to go into one of our 4 treatment groups, and we're going to follow them for up to a year while we're treating them. These are the collaborators we're working with, again, looking at a variety of tissue factors from genomics and drug resistant. What makes those tumors spread? Can we detect this disease in the blood? Can we come up with a test that we can use in the emergency room to differentiate those guys with benign lesions from malignant lesions? Interestingly enough, we have found that many dogs have benign lesions. You know, we used to say it was only a quarter of them or a third of them that have benign lesions and the rest had cancer, but it's actually more like 60% with cancer, 40% without. So almost a coin toss. And so I think it's really important that we don't euthanize these guys without really looking to see what we have beforehand. And so that's really all I have for you today, given the amount of time that we have, but I'd be happy to answer any questions you guys might have about Ethos Discovery, our focus, what we're doing, or either of these 2 trials that we talked about today.
Okay, thanks everyone. I'm going to be joined out front here, the virtual front, the virtual stage with Heather and Dave. And I have a couple of questions that have come in. And so this is I'm going to try my best to get through all these questions, everyone. So hang in there with me. First, somebody asked about melanoma, and I don't know if either of you guys, either of your groups are looking at melanoma in dogs.
I'll just quickly, at the moment, that's not a big area of where we've been looking, either for SINE inhibitors or for bispecific. So at the moment, no.
We have enrolled a couple of dogs in the MIMIC study, which is the metastectomy study for dogs who have melanoma that has spread to their lungs. But we don't have an active study looking at new novel drug therapies. Our AOH 1996 trial will take all comers at the moment. And so that could include a dog with melanoma, but isn't specifically for melanoma.
Okay, I had a question, someone asked a question, and maybe you guys can answer this, and may have seen it come in about blue light cystoscopy. Do you know if anyone's using that in the veterinary space? I can't say we are on the Morse side, but I don't know.
I'll let the internist take that first.
Yeah. The only experience we have is that we did some work with Cedars-Sinai looking at chemicals that we could inject peripherally that would differentiate brain tumors from normal brain tissue at the time of surgical removal. But we haven't done anything with fluorescence markers or anything related to surgery.
Yeah. So I don't do much cystoscopy myself. I know that a lot of groups are doing cystoscopy with laser ablation and things like that. But as far as blue light cystoscopy goes, I'm not very familiar with groups who are using that for UCC or bladder tumor treatments in dogs. But I think we can keep an eye out and see, but I think a lot of people are using more local therapies like laser ablation and things like that.
Okay. I'm going to take one here. I'm going to share it with you guys, but someone asked, are there any studies in progress, genetic studies, to see why Goldens are more susceptible? And I am going to tell you guys that's a big part of the Golden Retriever Lifetime Study. Obviously, we're looking at lots of data. We have samples. We have those available for researchers, but I'm also going to toss it to our panel today. Are you guys doing anything specific in the genetic piece?
So we have a huge genetics component with a lot of the work that we do. A lot of it is understanding the genomics of the specific disease as opposed to the breed associated risks. That being said, we do look at all the different breeds as they come in, and we do occasionally find differences in some of these breeds. But for the most part, the genomics work that we're doing is truly disease-based as opposed to breed-based genomics. When you get sort of post-diagnosis, the disease genomics are probably a bit more important. Pre-diagnosis, the breed genomics may be more important. It may also be something that the Dog Aging Project is able to look into, and that may be another group in addition to Morris, who actually is truly golden focused as opposed to the Dog Aging Project, but they may also have some data that hopefully will come out at some point as well.
Okay, here's another big one, mast cells. Anything going on with both of you guys with mast cell tumors?
Well, we've looked at mast cell tumors, I think, from a drug pharmaceutical development standpoint. Most of the mast cell tumors are dealt with surgically. Fortunately, most of them are not life-threatening tumors. For the more aggressive ones, there have been some work done with, obviously, with kinase inhibitors. And I think there's some pretty good human models for looking at systemic mastocytosis and maybe some of those drugs being repurposed for mast cell.
Yeah, I don't have any open trials for mast cell tumor right now.
Yeah, and I'm going to put both of you a little bit on the spot because I think there may be folks who aren't as familiar with mast cells. So can you give sort of the 30,000-foot review of mast cell tumors in dogs, because they're kind of weird and they fall into different camps, right?
That's
Sure.
an oncology question. Yeah.
So mast cells are normal in your body, and they're part of your body's normal allergic response or a response to parasites. And so they're everywhere in your body because they're part of that sort of immune surveillance for allergies and things like that. So when you get a mosquito bite, the part that makes it really itchy is sort of your body's response to the saliva from the mosquito, not the mosquito saliva itself. And that's sort of what your mast cells are doing. And so when you get a mast cell tumor, they most commonly occur in the skin or just under the skin in the subcutis. And we find that dog breeds with short noses tend to be predisposed. So your boxers, bulldogs, Bostons, pugs, those kinds of guys. We can see it in any dog. It's so common that no breed is spared, but those short-faced dogs tend to get them more often. In many cases, so there's sort of two main grades. We kind of consider them high grade or really aggressive and low grade. The high grade tumors are uncommon, but when they are high grade, they are very aggressive. The vast majority of these are low grade tumors, which actually can often be cured with surgery if they're in a good location, and maybe need some chemo depending on where they are or some of the factors that we see when we send them to the pathologist and get that report back. High-grade tumors, we rarely cure, and most of the time have to treat with chemo and follow pretty aggressively. And so when those tumors spread, they don't usually spread to the lungs. They like the liver, they like the spleen, they like lymph nodes, and they can make dogs feel sick, vomiting and things causing gastrointestinal ulcers. They can also get bad enough that they can cause actual collapse episodes, almost like an anaphylactic or a severe allergic reaction. So those are that's the maybe quick summary. I don't know if that's
It's perfect. No, that was great. I have a question here, and it is so perfect for Dave. It is like somebody will use a golf reference. They've put it on the tee. They've lined it up, the shot, or volleyball to spike. Someone asked about research on valley fever. So I think Dave can talk about this and their new product.
Yeah, I mean, we've been active in valley fever for about 5 years now. We're bringing forward a vaccine through the USDA to prevent valley fever. We finished the efficacy challenge model studies. We did those with the folks at Colorado State. Finished our safety studies as well. So right now we're getting ready to submit some additional data to USDA prior to doing our pet dog field study. And the pet dog field study will be sort of the last clinical bit that we have to complete in order to file for regulatory approval. So we're hoping that if all goes well, that there'll be a vaccine available in 2024.
That's fantastic. For those of you living on the East Coast, I didn't have any idea. I'm a Jersey girl, so I never heard about valley fever till I came to Colorado. And even then, we didn't see it. It was only people who traveled, maybe had a home in the Arizona area, right, or California would travel. So this is but it's a big problem. So it's very exciting. Someone asked about a particularly nasty cancer, histiocytic sarcoma. So I'm wondering if any of you guys are doing anything with that.
We're not active in looking into that, so I don't know. Heather may have some better insights.
Yeah, so histiocytic sarcoma is a really tough disease, and we don't have a lot of good options. I mean, it's one of the few diseases that's probably as bad, if not maybe in some cases worse than hemangiosarcoma. We don't have true active areas specifically in histiocytic sarcoma, but it's fortunately not nearly as common as lymphoma or hemangiosarcoma. So I know it is an interest because there are some clear genetic mutations that are associated with those, but we don't actively have any trials going on right this minute with that one.
Yeah, it's a tough one. I'm going to put in a small commercial for, again, the Golden Retriever Lifetime Study, because much to our surprise, it is the third most common cancer we're seeing in our group. We anticipated hemangiosarcoma and lymphoma. We did not anticipate that, again, histiocytic sarcoma would exceed osteosarcoma or high-grade mast cell tumor. It's way up there. And I'm going to speculate the reason is because I don't want to say we force people, but a lot of people undergo biopsy or a postmortem examination. I think we find, and Heather can comment on this some more, I think we find histiocytic sarcoma when we think it's hemangiosarcoma, right? And so people, I will be the first to own, when I was in practice, if I saw something that looked like it and we elected to euthanize the pet for a variety of reasons, I wouldn't maybe go in and get a piece of tissue to find out. And for the people listening, because of our great samples and data, we are hoping that we can entice people to look at histiocytic sarcoma in a way that may not have always been available before. But it was surprising, and it's as Heather mentioned, it's a pretty nasty kind of tumor. I have another question, and this is outside the purview, I think, of both your companies, but you can talk a little bit about it. Liquid biopsy for detection of cancer early. What are you guys doing? What do you know about it?
Go, Heather.
Yeah, so full disclosure, I am a paid consultant for Volition Veterinary, which is a liquid biopsy company. So I will just put that out there. I will say that I think it is a hot topic. I think it's an area that people are really interested in and really getting into. I think, we want what we see on the human side a lot, which is a blood test, like a PSA that can tell you, do you have cancer or not? And if you do, can we use that for monitoring or things like that? As an oncologist, I'm personally most excited about the ability to use these tests for monitoring purposes, because that's what I do. I treat these cancers every day. I want to know if my treatments are working. I want to know if it's coming back earlier, and those types of things. There's a variety of different types out there. There's some that are more specific, that use next generation sequencing. They take a little longer, they're a little more expensive, but they are more accurate. There's some that are a little less accurate, that are more global, looking at total kind of circulating DNA, cell-free DNA, which kind of helps you. If that's really high, is an indication potentially of cancer. Those are cheaper, but not as specific. And so there's sort of the whole mix and the gamut. I think I think in breeds like Goldens that are at risk for cancer, such high risk, I do think there's an opportunity there for some screening, or at least cancer monitoring associated with those breeds. So I think we're at the dawn of our genomics era in cancer research and veterinary medicine, and my hope is that they will only get better and better, and that we'll be able to really rely on some of these tests, or some of these companies who are coming out with these tests to really help us in a non-invasive way detect and monitor these diseases in our pets.
Yeah, I think that's something that we're hoping, and I can share with the audience. We have a couple researchers who, of course, have asked for our samples, right? Because we might have a sample 2, 3, 4 years before a pet is diagnosed with lymphoma and hemangiosarcoma are the two that I'm thinking of. So we do have a collaborative effort, at least in lymphoma, where they're looking at, first they're testing sort of a proof of concept, like we think this is a marker. So we have to give them some samples that dogs never had lymphoma, dogs that did. And we also share maybe the 3 years leading up to the diagnosis, when to get at that idea that Heather mentioned, which is, if we're going to screen these guys, well, how often do we need to screen them? When could we maybe anticipate that we could detect something in a test? So that's really exciting stuff. And we're a ways away. I don't want to lead anyone to believe like this is going to be coming out in the next 3 or 4 months, but we are heading there. And a lot of people, I think, are heading there. So I've got one more question for you guys, and then we'll wrap it up. It says, okay, if you have a dog with hemangiosarcoma and do you usually see metastasis to other organs, which would make spleen removal a less favorable treatment outcome and a more positive outcome for the pet owner? So maybe, I don't know, maybe Heather, you want to tackle it as the oncologist, but.
Yeah, so the thing about hemangiosarcoma is it doesn't just have a high rate of metastasis, it spreads early in the disease process, right? And that's different from osteosarcoma, which they often come in with a primary tumor, but they rarely have spread at the time of diagnosis. So hemangio, that does happen pretty often. In many cases, we will do our best to screen these guys before we take them to surgery when they come in through the ER, but we may not always find the evidence of spread until we get into surgery in those cases, but we'll do chest x-rays and things to look for that. Certainly, we kind of have 3 stages of lymphoma. Most cancers have 4 stages, but hemangiosarcoma is just so awful it stops at 3, I guess. But the stage 1 are these small tumors that haven't bled yet. They're less than 5 centimeters. Those dogs absolutely have a better prognosis when we take them to surgery. So if we were to find that incidentally on ultrasound, for example, because they had, some vomiting, or they got in the garbage can, or we were doing it as part of their wellness exam, those guys have a much better prognosis than the ones who have come in and they're stage 2, which means it's bigger than 5 centimeters, or it's already bled, or there might be some spread to the areas, like to the lymph nodes or things right around that primary tumor. And then stage 3 are those guys that have spread, sort of beyond that cavity. So they may have a splenic mass, but it's spread to the heart or the lungs, or vice versa. And those guys definitely have the shortest survival times. When we do find that there's broad evidence of spread, we do find that those guys do not get as much time after surgery. Stopping the bleeding is sort of a triage method to kind of get them through the night and to sort of get hemostasis, or control that shock situation, that emergent situation, but they still have measurable disease that's going to continue to bleed, continue to spread, which makes that those guys definitely have a worse prognosis. So our goal is always to try to catch that disease earlier if possible, so that we can treat them earlier before the diseases become very broad spread.
Okay, I know we're a little over, but Dave, I'm going to ask you one final question, which is, someone asked about if their dog can participate in the Valley fever vaccine trial, and if you're recruiting.
Yeah, certainly if you live in Arizona, I would just reach out to your local veterinarian. We'll be setting up trial sites. And so, yeah, we would love to have your dog be a part of that.
Great. All right, we are at 101. I want to thank Dr. David Bruyette and Dr. Heather Wilson-Robles for joining us, and a really informative and wonderful lecture. And we will be posting this probably within a week. So if you have friends who missed it, we'll be sending a link. If there's something you want to look at again, you'll be able to do that. We'll have some resources in the resource section with the video. It'll probably be a couple links to some other stuff we're doing. And that's it for us. So again, thanks to everyone for joining us today. We really appreciate it.
Thank you.
Thank
Take
you.
care.